Well, hello, everybody. This is going to be the first half, well, a little bit more than half, actually. First part of chapter 14, pages 473 to 47. And just like last time, this one's probably going to be on the longer side, hopefully not very long. And then the second video this week will be on the shorter side. It just doesn't split evenly in half. I'll also add that I've been waiting to do this lecture for pretty much the whole semester. There are a few key lectures, a few key topics that really are my favorite in cell biology, and what we're going to talk about today is most definitely one of my personal favorites. Really, really interesting and fascinating story. So what are we going to talk about? Well, we'll start off by just giving ourselves some idea of the anatomy of the mitochondria and why mitochondrial membranes are so important. We'll discuss and define the process that's referred to as chemoosmotic coupling, joining a chemical gradient with an osmotic process. Again, that'll circle us back to the mitochondria. And then we'll pick up where we left off on the citric acid cycle. We've harvested electrons, a lot of them on NADH and 2 electrons on FADH2. We'll remind ourselves where those electrons came from and then talk about where they go, which is the electron transport chain. We'll talk about how the electron transport chain really works to pump protons against their concentration gradient, which will lead us to the magnificent and amazing enzyme ATP synthase and how ATP is made. And we'll close off by talking about the overall efficiency of this process being aerobic respiration, the release of energy from glucose and the production of ATP done in the presence of oxygen. So, how is ATP generated in eukaryotes? Before we took our last little video detour into gluconeogenesis, glycolysis regulation, and the production of glycogen, before we did that, we had left last week's material at the end of the citric acid cycle. Glucose had gone through glycolysis, pyruvate was converted into acetyl-CoA, and that acetyl-CoA was fed into the citric acid cycle. And in the citric acid cycle, all of the bonds of glucose were broken down, releasing the carbons of glucose as carbon dioxide. Yes, we made a little bit of ATP along the way, 3 triphosphates in total, but the purpose of all of that was to steal all of those electrons, and we stole a lot of them. Remember, those electrons that we stole were the high energy electrons that were harvested from the covalent bonds of glucose. The covalent bonds of glucose contain an enormous amount of potential energy. That potential energy is in the electrons of those bonds, and it's those electrons that we stole pretty much in the citric acid cycle. Now those electrons can be passed from an electron carrier to an electron carrier through subsequent redox reactions. It's almost like the electrons are a hot potato and they're being thrown from carrier to carrier. And all of those carriers today will exist along the inner mitochondrial membrane. And as the energy of those electrons is slowly released with each spontaneous redox reaction, with each redox reaction that has a negative delta G, a little bit of that energy from the electrons will be released. As that energy is released, we'll capture that energy, we'll use it to pump protons, and we'll use that proton gradient to make ATP. And this is the process of aerobic respiration. When we're done with those electrons, we'll see that we use oxygen to get rid of them, which is what makes this process aerobic. Aerobic respiration was first evolved by prokaryotes, and still to this day, it's only been evolved by prokaryotes. Remember, since this is happening in the mitochondria, this is actually still happening in prokaryotes even today. The prokaryotes that our cells ingested a long time ago but never digested, the prokaryotes that ultimately became the mitochondria. So what we're doing is we're making ATP using and across a membrane. That's what the mitochondria's power really is its internal inner membrane and the proteins that exist within and along that membrane. The point of everything we're doing here is to make ATP, this universal energy molecule of life. And the ATP that we made through glycolysis and that single triphosphate we made in the citric acid cycle, it's enough to keep very simple cells living, cells that ferment for most of their lives. It's also enough to keep muscle cells alive for a little bit as oxygen becomes deprived in our own body. But the amount of ATP made during glycolysis in the citric acid cycle is not nearly enough to sustain the complexity of eukaryotic life over an extended period of time. That complexity of our life requires a lot of universe bribing. We are very anti-entropy in our physiology. And in order to release enough heat to bribe the universe to allow eukaryotic life to be possible over a long term, we have to have a lot more energy harvested from glucose and convert that energy into ATP. A lot of ATP is needed. And that's what oxidative phosphorylation is for. It's for making these high levels of ATP from glucose. Oxidative phosphorylation is the phosphorylation of adenosine diphosphate to adenosine triphosphate using energy derived by oxidation. Well, what's oxidation? Oxidation is a reaction that is electron stealing. When you oxidize something, you've stolen electrons from it. So the very name of this process tells you exactly what's going on. ATP is being made using energy of oxidation, using energy of electron theft. And OxPhos is highly efficient. It is the most efficient way to capture the vast majority, well, I shouldn't say the vast majority. It's the most efficient way to catch about half of the energy of glucose and use about half of the energy of glucose for ATP synthesis, but that is hands down and by far the most efficient capturing we are able to achieve. But for OxPhos to be possible, we need these membrane-bound compartments. We need proteins for redox reactions along a membrane, and we need protons to be able to be pumped across a membrane. And in eukaryotic cells, the source of these membranes for OxPhos to be possible is the inner lining of the mitochondria. Electrons are transferred across carriers on the inner membrane, and protons are pumped against their concentration gradient across the inner mitochondrial membrane. And we see that here, represented simply. We're going to go into this in much more detail, but we see here that first, the high energy electrons that we've harvested and stolen using the citric acid cycle arrive at the inner mitochondrial membrane, and those electrons are offloaded or passed onto the electron transport chain. And that's what we see right here. These high energy electrons being passed through the electron transport chain. And then electron transport chain carrier to another, the energy from those electrons is released and directly used for the active transport of protons. Protons being pumped across their concentration gradient from low to high. That's active transport. Each one of those redox reactions that allows these electrons to be passed from carrier to carrier of the ETC, all of those reactions are spontaneous. They have a negative delta G. They're releasing energy, and some of that energy is used to pump these protons against their concentration gradient. The rest of that energy, of course, is released as heat to bribe the universe, but what we're after is this proton gradient. Once we've created this proton gradient, we've got a high concentration of protons on one side of the inner mitochondrial membrane, and we have a low concentration of protons on the other side. Well, now we have a proton gradient. We have energy of want. We've taken the potential energy that was in the bonds of glucose. We've robbed that potential energy as potential energy trapped in the electrons of those bonds, and now we've converted that potential energy into the potential energy of want of this proton gradient. These protons desperately want to flow from high to low, and that is energy of want. That's potential energy. When those protons are allowed to flow, that potential energy becomes kinetic energy, and those protons will flow through a proton channel protein. A proton channel protein that's embedded within the mitochondrial membrane. And that channel is part of ATP synthase. As those protons flow, they will cause this ATP synthase channel to physically spin. There's your kinetic energy. And this kinetic energy of ATP synthase spinning is the energy that will be used to phosphorylate adenosine diphosphate to adenosine triphosphate. The torque energy of ATP synthase spinning is the energy that can grab one more negative phosphate and jam it onto two negative phosphates, creating that high potential energy of repulsion that we find in ATP. So really what we're seeing here is two energy conversions: the potential energy from the electrons of glucose to the kinetic energy of protons flowing to the chemical energy of ATP bonds. ATP synthase is a turbine. It creates energy through spinning, just the way a hydroelectric plant does that's based in the dam over some kind of bridge or waterway. So what we've been talking about here, whether we realize it and appreciate it or not, is this idea of chemoosmotic coupling. Chemoosmotic coupling is the direct linkage between chemical bond forming energy needed to make that triphosphate bond of ATP. So basically the chemical bond forming energy of ATP synthesis. That's the chemi part, making a chemical, with the potential energy that came from the concentration gradient of protons trapped on one side of the inner mitochondrial membrane. And that's that osmotic piece. Usually we use the term of osmosis for water flow. We're kind of maybe inappropriately borrowing that term here because it's not water that's flowing, it's protons that are flowing, a component of water. But the idea is the same. Chemoosmotic coupling is taking the osmotic pressure, the osmotic potential energy of a proton gradient and linking it directly to the production of chemical energy in ATP. That potential energy of want to flow is actually a fundamental premise of all trapped energy. Batteries, even the batteries that we use in our everyday lives, function on this same principle. Every battery has a negative end and a positive end, and electrons want to flow from negative to positive, but they can't because they're on opposite sides of the battery. So you have potential energy of current there, potential energy of want. That's what we call a voltage. Now, if you connect a wire or a circuit to both ends of the battery, well, now the electrons can flow, and that potential energy of wanting to flow is converted into kinetic energy of electron flowing. If now you take a light bulb and you put it between those wires, well, as the electrons go through the light bulb, they're going to have some resistance, some friction, and that's going to make the light bulb glow. Or if you put a pump between those wires, now as the electrons move through the pump, they're going to cause the pump to operate, and we can pump water from one place to another. So a battery functions on the principle of potential energy of wanted flow. And then once those electrons are allowed to flow, that energy goes from potential to kinetic, does work. Same idea in a hydroelectric plant. On one side of the dam, you have a lot of water. On the other side of the dam, you have relatively little. That water wants to flow. You have trapped potential energy of wanted flow. But no power is generated unless you open the dam gates. DAM gates, not DAMN gates. We're keeping this PG. Once you open the dam gates, the water will flow through the turbine and cause the turbine to spin. And now that kinetic energy of water flowing is used to generate electricity. And the same principle is happening here. We're going to create a proton gradient. We're going to create wanted flow energy, potential energy of wanted flow, and we can tap into that energy and turn it into kinetic energy once those protons are allowed to flow. This incredible process of taking the energy from glucose, stealing it through oxidation reactions, using it to pump protons, and then harvesting that proton potential energy to create ATP, this process evolved in prokaryotes over 3 billion, billion with a B, billion years ago, and still even today, it's only prokaryotes that can pull this off. Don't let yourself forget where mitochondria came from. Don't forget the endosymbiotic hypothesis. Mitochondria were nothing more than prokaryotes that were consumed and engulfed by a larger cell but never digested. Chloroplasts too, same origin story. And these mitochondria still retain many of their prokaryotic origin traits. They still have their own circular genome. They're often still modal, and they still perform this aerobic respiration trick. They replicate, they have their own DNA, their own RNA, and even mitochondria are the only eukaryotic component that's capable of aerobic respiration, making this still a, if you want to think about it, exclusively prokaryotic process. Now, to be fair, mitochondria are not prokaryotes anymore. You can't harvest mitochondria from a eukaryotic cell and grow it on a petri dish or grow it in culture. Mitochondria now depend on the larger cell for survival, but make no mistake about it, we depend on them as well. They need us as much as we need them, and we need our mitochondria as much as they need us. And so let's talk a little bit more about mitochondria specifically, because this is where oxidative phosphorylation is occurring. Mitochondria are present in nearly all different types of eukaryotic cells, and they make by far the vastest amount, percentage-wise, of the ATP being made by eukaryotic cells. Without mitochondria and the oxidative phosphorylation that they allow, it is a fact that eukaryotic life would not be possible. We're simply too complex and require too much ATP for us to get by without aerobic respiration. we make, what did we say, about 3 triphosphates with the citric acid cycle and glycolysis. Well, we make 30. We're going to see 30 ATPs with OxPhos. So quite literally, oxidative phosphorylation is 10 times or more efficient at extracting the potential energy from the covalent bonds of glucose and releasing that energy to make ATP. Interestingly, in some cells, mitochondria aren't modal. In some cells, they're stationary and they just grind out ATP and make tons of ATP exactly where it's needed. Some examples of this is in cardiac muscle cells. The mitochondria are stacked between the actin myosin muscle fibers that are actually responsible for the contraction. So ATP is being made and dumped and delivered exactly on the components of the cardiac muscle that are responsible for keeping the heart beating. Also, in the tail of a sperm, mitochondria actually self-organize and they connect into this spiraling, almost coating that wraps around the motor end of the flagella and just dump ATP into the flagellum motor to keep those sperm cells swimming. So these are examples of stationary mitochondria. But in most cells, mitochondria are fairly fluid and dynamic. They evenly distribute across the cytoplasm. Sometimes they connect into this large continuous mitochondrial network. Sometimes they actually expand as a single mitochondria and take on a very large size. Sometimes they remain our typical kidney bean shape and we just have many, many of them in a cell. But in all cases, they are what's doing aerobic respiration. They're making the ATP. A relatively small number of mitochondria in a cell is 1 to 2,000. Liver cells have 1 to 2,000 mitochondria, and again, that's fairly paltry. And we can get to 10 times that number in cells that require a high level of ATP, like muscle cells. So we would see 10 to 20,000 mitochondria in each single muscle cell because muscle cells do so much work. They require so much energy for the movement that they allow. And I also just want to take like, what, 3 minutes maybe, and give a little bit of appreciation to other things that mitochondria do, because they've too much more than just aerobic respiration. Mitochondria are responsible for making the molecular precursors for a lot of macromolecules, especially some fats and some amino acids. So the mitochondria make those. In addition, mitochondria are responsible for making heme that's needed for hemoglobin in the red blood cells. And apoptosis begins, or the recognition of the need for apoptosis and the early stages of apoptosis occur when mitochondria release certain toxic compounds that ultimately cause the cell to die. So table 14.1 in your textbook gives this appreciation. And yes, ATP production is the primary purpose of mitochondria, carrying out oxidative phosphorylation and aerobic respiration. And when you do OxPhos and aerobic respiration, you're regenerating NAD+, so you don't need fermentation anymore. You're offloading your electrons onto the electron transport chain. NADH is being oxidized back to NAD+, and these cycles can continue. Super important, most important, definitely belongs at the top of the list. But again, we're making a lot of precursors for biosynthesis, like I mentioned, certain amino acids, some nucleotides, some fatty acids. We're making heme. Cell signaling is mediated by some mitochondrial components, and then we're generating reactive oxidative species that allow a cell to sometimes carry out certain processes, but other times just initiate apoptosis, and other apoptosis triggering events also occur in the mitochondria. So let's get to some mitochondrial anatomy. The deepest part of the mitochondria, if you were to go through all of the membranes that mitochondria have, and you're at the most internal part of the mitochondria, that's called the mitochondrial matrix. It's an empty space, well, not empty. It's got its own cytoplasm kind of stuff in there. It's got fluid in it, but there's no membrane. There's no structure in there. Mitochondrial matrix is a privileged space. Access to the mitochondrial matrix is tightly regulated. Then we have the very thin kind of limbo land between the inner mitochondrial membrane and the outer mitochondrial membrane. That's that very thin area that we see right here. This thin limbo land is called the intermembrane space. And the intermembrane space is quite empty. There are some proteins here. There's some regulation of what can go deeper into the matrix occurs here. And certainly all of the proteins of the ETC, of the electron transport chain, are associated with or embedded into the inner membrane itself. But the intermembrane space between the two membranes, not too much happens there. And then we have the outer membrane, the surface of the mitochondria. And this is largely protective. It contains transport proteins that are needed to get things into the mitochondria. Most notably to us at this point is pyruvate, right? Pyruvate is made by glycolysis in the cytoplasm, and then pyruvate is transported into the mitochondria to be converted to acetyl-CoA. Well, that means pyruvate needs to get through this outer membrane. And so the outer membrane is like a gatekeeper function. The proteins in the outer membrane that allow things to enter the mitochondria are called porins. They're channels, usually, transporters. And we can see the green arrows here are representing some of those porins. So metabolites are getting into and out of the outer membrane, into this intermembrane space. And then most of those molecules are very quickly grabbed by additional porins that are in the inner mitochondrial membrane and brought into the matrix. So this region here would be the matrix. This region here would be the matrix. This is the inner membrane, intermembrane space, outer membrane. The outer membrane is unguarded, meaning the passive diffusion across that membrane isn't nearly as restricted or impermeable as it is for the inner membrane, but it's a membrane. So if you're charged, you can't get through. If you're very large and partially charged, you can't get through. Everything you know about the lipid bilayer applies to the outer membrane of the mitochondria as well. Oxidative phosphorylation, the process itself, and making ATP, that occurs at, along, and across the inner mitochondrial membrane. The proteins of the electron transport chain are embedded into the inner mitochondrial membrane. ATP synthase spans the inner mitochondrial membrane. Protons are going to be pumped across this membrane. Protons flow through ATP synthase across this membrane. All of the magic of aerobic respiration and oxidative phosphorylation occurs at the inner mitochondrial membrane. And because of that, because ATP production is so important, and ATP production quite literally and spatially happens at this inner mitochondrial membrane, the inner mitochondrial membrane is articulated. It's folded and convoluted upon itself, as we see here, these kind of switchbacks and convolutions in a way to increase the surface area of that membrane as much as possible. The more inner mitochondrial membrane you have, the more electron transport chains and ATP synthases you can have, the more ATP you can make. So it's the same exact reason why the brain has the structure that it does. All these convolutions, these folds, this texture creates much more surface area, and therefore more can get done. The more inner membrane you have, the more ATP you can make. It comes down to that. These structures, these convoluted structures, due to the infoldings of the inner membrane, are referred to as cristae. And I like to think of them instead as that word kind of mean creases, right? Like creases are folds in your clothing. Cristae are the folds of the inner membrane of the mitochondria. All right, so we've kind of built a pretty strong foundation here. Hopefully we're comfortable with where we are in the mitochondria and what's going to happen from a big picture perspective. Let's start filling in the details. Let's go back to the citric acid cycle where we left off last week. And remember that all of the energy producing, high potential energy, high energy electron containing foodstuffs that we've consumed are usually digested down into their monomers and then ultimately converted into acetyl-CoA. For sugars, that's going to be polysaccharides digested into monosaccharides, monosaccharides converted into glucose, glucose converted into pyruvate through the glycolysis, and then pyruvate imported into the mitochondria so it can be converted into acetyl-CoA. For fats, that's going to be triacylglycerides broken down into 3 fatty acids and glycerol. Then those fatty acids are going to be broken down into smaller hydrocarbons via a process called beta oxidation. And then those hydrocarbons will be transported into the mitochondria and converted into acetyl-CoA. Same kind of thing's going to happen with the carbon backbone of a lot of amino acids. They'll be digested in the digestive system down into individual amino acids. And then individual amino acids will be broken down into their carbon skeletons in the cytosol. And then those will be transported into the mitochondria to be converted into acetyl-CoA. So acetyl-CoA is made from 3 of the 4 macromolecules. It's made from fats, from sugars, and from amino acids, from proteins. But the actual production of acetyl-CoA is mitochondrial. It occurs in the mitochondrial matrix. And all of these things are getting into the mitochondrial matrix by going through these porins of the inner mitochondrial membrane and the outer membrane as well. Once acetyl is made, it's going to be catabolized by itself. Its bonds are going to be broken. Its electrons are going to be stolen. And all of that is happening by the citric acid cycle. That's what the citric acid cycle does. Acetyl is linked to oxaloacetate, making citrate. And then citrate, step by step, is slowly decarboxylated. And bonds are broken in that decarboxylation. Electrons from those bonds are stolen and used to reduce NAD to NADH or FAD to FADH2. So the citric acid cycle breaks these sugars, these fats, or these proteins down basically into electrons, electrons that can be used to with the electron transport chain. So as we break these bonds and we do these oxidations, we steal these electrons, what we're really doing are redox reactions. The oxidation reactions are stealing the electrons from acetyl, and the reduction is putting those electrons onto the carriers. Usually the carrier is NAD, and sometimes it's FAD. These carriers are then used to deliver those electrons to the electron transport chain. What we see here in the lower right is how NAD is actually doing this. We see NADH on the left. It's carrying its two high energy electrons right up there. That's giving that carbon the ability to make another bond, and it's making that bond with the H. That's why NAD plus goes to NADH. And as NADH with its electrons and its proton arrives at the electron transport chain, it's going to offload both. It's going to offload the proton. Well, in fact, that's going to be some of the protons that are pumped. And it's going to offload the electron because that's where the energy is. And the electron will be passed to something else that's part of the electron transport chain. And that also is a redox reaction. In this redox reaction, it's NADH that's being oxidized and the electron transport chain that's being reduced. So electrons are being passed from carrier to carrier through redox reactions. Oxidation is gaining the electron. Oxidation is stealing the electron, and the reduction is putting that electron on a carrier. And then we continue to link these redox reactions together, and we pass the electrons like a hot potato. So I titled this slide, "Why are these electrons high energy?" And as I was going through these slides, getting ready to make the recording, maybe it shouldn't be why are they high energy, but like why are we saying they're high energy would probably be a better title for this slide. The electrons are high energy because they were involved in making the covalent bonds of a sugar, an amino acid, or a fat. And in those covalent bonds, like stretched out rubber bands, there was a lot of potential energy. So they're high energy electrons because they were electrons used to make covalent bonds, and covalent bonds contain a lot of potential energy. That's why they're high energy. But why do we get to say they're high energy? Or what do we mean when we say high energy? Well, that's what I'm trying to get at here. Let's take a look at what's actually happening here. What we're seeing is a chain of redox reactions. NADH is in its reduced form, and it loses its electrons. So NADH becomes oxidized, but it loses its electrons to FAD. FAD becomes reduced. FADH2 is going to lose its electrons. It's going back to being oxidized, but it's going to give its electrons to this beta complex. The beta complex gets reduced. The beta complex is going to lose those electrons. It's oxidized. It's going to give it to a cytochrome, reduced. Cytochrome is going to lose those electrons. It's getting oxidized. It's going to give it to complex A, reduced. Complex A is going to lose those electrons. It's oxidized, and it's going to reduce oxygen. And when oxygen is reduced, remember, follow the protons. When oxygen is reduced and it gains electrons, it becomes water. We'll come back to that in a second. So this is the game of hot potato. These are redox reactions linked together, and electrons are being passed down a chain. And each time we do one of these redox reactions, look what we get. The delta G, big and negative, love it. Small and negative, but still spontaneous, big and negative, small and negative, huge and negative, which each of these passings of the electrons, which each of these redox reactions, we're losing energy in these spontaneous reactions. We have negative delta Gs. Well, where did this energy come from, right? What do you mean we're losing energy? What do you mean a negative delta G? Where did that energy come from? It came from the electrons. See, the electrons were very high in energy when this started. The electrons that NAD are carrying are high energy electrons. They are the electrons from the covalent bonds of those monomers. And then as we do these changes, as we pass these electrons on, we'll oxidize NADH and reduce CoQ. We'll oxidize CoQ and reduce cytochrome B. We'll reduce cytochrome B and we'll oxidize cytochrome C. We'll reduce cytochrome C and, I'm sorry, we'll oxidize cytochrome C and reduce cytochrome A. Again, as we do each of these subsequent redox reactions, the amount of energy in the electrons gets lower and lower and lower. Why? Because it's being released by each of those spontaneous redox reactions. The loss of the electron energy we see here as we go down the y-axis is represented by these negative delta G's of energy release here. And so redox reaction by redox reaction, step by step of the ETC, we're releasing energy from those electrons, and that energy is coming out as free energy of those redox reactions until ultimately there's no energy left on those electrons at all, and then we put them on oxygen and get rid of them as trash, as waste. Okay, great. So we're releasing energy as these electrons are being handed off from NADH or FADH2 to the complexes of the ETC. And as those subsequent redox reactions are occurring, each of them spontaneous, each of them releasing energy, as that's all happening, free energy is being released for what? Well, for proton pumping. We're pumping protons against their concentration gradient. We're taking protons from a low concentration side and pumping them across the inner mitochondrial membrane to a high proton concentration side. And they don't want to do that. That's against their gradient. So whenever you pump anything from low to high, you're doing active transport. And as you've learned in other courses, I'm sure, because for many of you, I'm the one who taught it to you, active transport requires energy. Well, the energy for active transport here at the inner mitochondrial membrane is this energy. It's the energy released from these redox reactions. And where did this energy come from, this energy that was released in the redox reactions? It came from the electrons themselves. So as these electrons are passed through the ETC and the energy from those electrons are released as spontaneous reactions, a lot of that energy is used for active transport of protons. Not all of it. Some residual energy is released to bribe the universe. It's released as heat to make the universe happy. But most of that energy is used, or at least half of that energy is used to create this proton gradient. The electrons are passed again to another complex of the ETC. We have a negative delta G. We've released energy. We're going to use some of that energy to pump protons, and we're going to release the rest of it as heat. And then we're going to pass those electrons to the next member of the ETC. We're going to have a high negative delta G. We're going to use some of that energy that was just released to pump protons, active transport, and the rest to bribe the universe. We're going to do that again and again and again until, again, we will have milked and released every last bit of energy out of those electrons that we can. And then with the last little tiny bit of those energy in those electrons, we'll give those electrons to something that loves electrons, to an atom that's highly electronegative anyway, to an atom that wants electrons even if they're energy poor. And that atom is, of course, oxygen. The electrons go on to oxygen. Because oxygen is accepting these electrons, oxygen can now make additional covalent bonds. It's going to make those covalent bonds with protons, 2 protons, in fact. And you take 2 protons and an oxygen, you just made water. And what's left from glucose is the proton gradient. Now take a minute to appreciate that. When we are done with everything we've just discussed, we took 6-carbon glucose and we converted it into, well, first pyruvate, then acetyl, where we lost some carbon dioxides, then harvested electrons in the citric acid cycle, where we lost the last of the carbon dioxides, and then we milked out all of the energy from the electrons possible, and we put the electrons from glucose onto oxygen, creating water. At this point, nothing is left from glucose. The carbons of glucose have long been exhaled. They were exhaled by the end of the citric acid cycle. And the electrons from glucose have now been put on water, and that's being exhaled too, as metabolic water, the water that you see as water vapor on a cold day. All that's left from glucose now is a proton gradient. So take a deep breath in, and you're breathing in oxygen. But I want you now to think of that oxygen as empty trash cans, because that's all oxygen is, man. That's all it is. It's something where we can put our waste. And the waste we put onto and into oxygen are low energy, spent electrons from glucose that have given up all of their potential energy for the pumping of protons. We put those garbage electrons, those low energy electrons, those spent electrons like an empty battery, we put those electrons onto oxygen and now breathe out water. Well, that water is the reduced oxygen, the filled trash cans that are now holding our garbage, holding our spent electrons and the protons that came along with them. That's all the breathing is. We breathe in for the oxygen. We want those trash cans empty. And we breathe out water, trash cans full of electrons, and carbon dioxide, which are the actual carbons of glucose that we don't need anymore. So, as I said, what's left from glucose at this point is just a proton gradient. That's all we've got. A proton gradient held back by the inner mitochondrial membrane. Now, if we allow those protons to flow, they can flow through ATP synthase, make it spin, and that kinetic energy of motion is going to be captured and used to phosphorylate ADP into ATP. That's the oxidative phosphorylation, the phosphorylation of ATP using energy derived by the oxidation of electrons. And so very, very simply, the ETC, as simply as we can represent it, electrons arrive being carried by NADH. NADH gives up those electrons and they're passed through the chain, but ultimately those electrons end up on oxygen, giving us water, and NAD is recycled to NAD plus its oxidized form. And all of the energy that was in those electrons was used to make a proton gradient that oxidative phosphorylation tapped into in order to phosphorylate ADP into ATP. So the inner mitochondrial membrane is the most crucial key component of this process. The inner mitochondrial membrane is what's keeping the protons from flowing arbitrarily. It's holding the protons back in the gradient. The complexes of the electron transport chain are embedded into the inner mitochondrial membrane, and ATP synthase is in the inner mitochondrial membrane. The protons flow across the inner mitochondrial membrane by going through ATP synthase, and that's what allows us to make the ATP. No inner mitochondrial membrane, no ATP. The more inner mitochondrial membrane you have, the more you can do this. And that's why we have those cristae, those inner foldings. Okay, so time to go one layer deeper into the complexity. Let's talk about the actual protein complexes that make up the ETC. The ETC is made up of over 40 different individual proteins, but they are physically grouped and conceptually grouped into three major complexes, complex 1, 3, and 4. There are lots and lots of electron transport chains all across the inner mitochondrial membrane. I don't want you to think that this is happening just in one place. We see here are the complexes of the of the ATC, complex 1, complex 3, complex 4. Here's ATP synthase. But as this diagram shows, they fit in a relatively small patch of the inner mitochondrial membrane. You could have another ETC and ATP synthase here. You could have another chain of ETC and ATP synthase here and here and here. So the inner mitochondrial membrane is studded with many, many, many electron transport chains and ATP synthase proteins. Each of them, however, are referred to as a respiratory complex, a respiratory circuit. Each of the individual complexes of the ETC, complex 1, complex 3, and complex 4, consists of multiple electron-carrying proteins and molecules. They're actually membrane anchoring proteins so that they don't diffuse in the inner mitochondrial membrane, and a lot of more complexity that we won't even touch upon here. So let's meet complex 1. Let me formally introduce you to complex 1 by its more proper name. Complex 1 is better known as NADH dehydrogenase complex. What a great name, right? We've talked about dehydrogenases before. What do dehydrogenases do? They oxidize things. They steal electrons. So the NADH dehydrogenase complex accepts and steals electrons from NADH. That's why it has that name. So this is where the electrons from the ETC and glycolysis are being delivered. They're being delivered to complex 1. Complex three's more formal name is cytochrome C reductase. So it sounds like complex 3 puts electrons onto cytochrome C, and indeed it does. And then complex 4 is known as the cytochrome C oxidase complex. Well, now we know what that means. It's oxidizing complex C, so complex 4 is accepting electrons from cytochrome C. And we're starting to get a sense of the chain, of the C of the ETC. We're starting to get a sense of the hot potato redox game we're playing here. But let's see how it unfolds. Each complex of the electron transport chain contains metal ions and other chemical groups such as iron-sulfur clusters. Well, those contain metal because they're iron-sulfur clusters. But they contain metal ions and other chemical groups that can be oxidized and reduced, that accept and hold electrons. And there are also mobile electron carriers that are shuttles between the complexes. So CoQ, or quinone, shuttles electrons from complex 1 to complex 3, and cytochrome C, which we've already heard about as in the formal names of these complexes. Cytochrome C is the mobile shuttle that carries electrons from complex 3 to complex 4. Now, I keep saying complex 1, 3, and 4, 1, 3, and 4, and you're probably wondering, like, well, what about complex 2? What happened to complex 2? Well, here's a mind blower. Complex 2's more formal name is succinate dehydrogenase. And I asked you to put a pin in that phrase. I asked you to remember that phrase when I first mentioned it. That is one of the enzymes of the citric acid cycle. Succinate dehydrogenase is the enzyme that steals electrons from succinate and puts them onto FADH2. So those FADH2 electrons, they're never shuttled to the ETC. Succinate dehydrogenase does double duty. Succinate dehydrogenase is actually an enzyme of the citric acid cycle, but then it holds its electrons and delivers them to the ETC directly. Yeah. How does complex 2? Well, let's use its formal name. How does succinate dehydrogenase deliver its electrons to the ETC? Through the CoQ shuttle, just like complex 1 does. So complex 1 accepts electrons from NADH. NADH is oxidized, complex 1 is reduced. Where's my cursor? NADH is oxidized, complex 1 is reduced. Then complex 1 is going to release some of the energy from those electrons through redox reactions that occur within the complex, and a lot of that energy is going to be used to pump protons. When the electrons reach the end of complex 1, they're going to be offloaded onto CoQ, and CoQ is going to shuttle those electrons to complex 3. Complex 3 is going to have its own internal redox reactions that release a lot of that energy, negative delta Gs, and a lot of that energy that was released is going to be used for active transport of proteins. When the electrons reach the end of complex 3, they're going to be used to reduce cytochrome C. Remember complexes 3's name, cytochrome C reductase. Well, cytochrome C reductase, because complex 3 passes its electrons to cytochrome C. Cytochrome C is the mobile shuttle. It's going to bring those electrons to complex 4. Complex 4's other name, cytochrome C oxidase. Well, cytochrome C is going to give up its electrons to complex 4. Complex 4 becomes reduced, cytochrome C becomes oxidized, some internal redox reactions within complex 4, and then it's complex 4 that adds those electrons onto oxygen, creating water when the electrons are depleted. A lot of protons were pumped, a lot of energy was released, and electrons were passed all along the way. Even though it's not shown here, complex 2, succinate dehydrogenase, is still holding its electrons from the citric acid cycle, never gave them up to anything, they're still on FADH2. Well, succinate dehydrogenase as complex 2 is going to give those electrons to CoQ. CoQ will deliver those electrons to complex 3, and then the rest happens as I just described. So CoQ is the entry point of all of the electrons for the ETC. All of the NADH electrons arrive through complex 1, and all of the FADH2 electrons arrive as part of complex 2. All the electrons culminate on CoQ. CoQ carries them to complex 3 to cytochrome C to complex 4 to oxygen, making metabolic water. And again, with almost every single one of these redox reactions, we have a negative delta G energy is released, and that energy is used to pump protons against their concentration gradient. It's used for the active transport of protons, making this potential energy gradient. And getting back to some of the things we talked about, oh man, what, at the beginning of last week? This is also the idea of releasing the potential of energy of glucose in really small, discrete steps. We made a big point of saying if we were to release all of the potential energy of glucose in one shot, it would be too much energy to capture. The delta G would be enormous, and so much of that escaped energy would basically cook the cell. And this is what we mean by discrete packets of energy. Each of these redox reactions is releasing a little bit of energy from glucose, and that energy can be captured and used to pump protons. We already talked about the metabolic water. So again, breathe in, oxygen, empty trash cans waiting for spent electrons. Breathe out, water vapor. That oxygen is now reduced, carrying those waste electrons, the electrons from glucose that are no longer of any value. Finally, let's talk about how we make the ATP. Got ourselves a proton gradient, and that proton gradient has an enormous amount of energy of want, an enormous amount of energy of potential energy of wanted flow. We can think of this proton gradient as a pH gradient, right? Because pH really is a measure of proton concentration. And in fact, we can measure this experimentally. We can see that the difference in pH on either side of the inner mitochondrial membrane is about a 0.7 pH unit. And if you remember from chemistry, the pH scale is a log scale. So every 1 on the pH scale is an order of magnitude. So 0.7 difference on the pH scale is a huge difference in proton concentration. But I don't want to negate the fact that we also have a charge gradient, a voltage gradient, because protons are ions. More specifically, protons are cations. They're positively charged ions. So as we pump more and more protons onto one side of the inner mitochondrial membrane, that means that side of the inner mitochondrial membrane is becoming more and more positively charged. And like charges repel. So the more positive it gets on one side of the inner mitochondrial membrane, the more repulsive it is for further protons to go there. Protons want to go where there's negative charge. They're repelled from areas that are highly positive. And so there are actually two separate forces of want driving protons across this membrane. There's the concentration gradient, because all molecules want to flow from high concentration to low concentration. But there's also this charge or voltage gradient, where the protons want to go from where it's very, very positive to where it's less positive. And whenever we have these two forces of want, one charge and one concentration, we call that an electrochemical gradient. Electrochemical gradient. The chemical gradient refers to the concentration gradient, and the electro, of course, refers to the charge. Electrochemical gradients play a huge role in the nervous system, except they're gradients of sodium and potassium and chloride rather than a gradient of protons. But this idea of using charge and concentration to really drive something across a membrane is a common one in biology. So what we want to leave this slide with is the understanding that these protons really, really want to flow. There's a lot of potential energy here because they're desperately trying to get away from being so crowded, and they're desperately also trying to get away from where it's so positive. So let's introduce the big bad boy, ATP synthase. ATP synthase is going to take all this potential energy of want and allow the kinetic energy of flow to make ATP. So let's harvest this potential energy of want and make ATP from it. And that's what ATP synthase is for. Oh, the bears! I forgot to mention the bears. That's why I put it here, to remind myself. So, yeah, well, let me come back to the bears. Let me come back to the bears in just a second. So ATP synthase is this remarkable protein complex, this channel, this enzyme, all in one. It is a huge and huge and complicated complex that serves many different purposes all at once. And it's ATP synthase that allows the magic of oxidative phosphorylation to occur. This protein is as complex, I mean that in the complicated way, and as incredible and as ancient as a protein comes. Just unbelievable. It's like science fiction, the way this thing works. And we see ATP synthase or ATP synthase-like proteins in, of course, bacteria and mitochondria. We also see these in archaeal cells. We see these in protists. We see these in chloroplasts. Anything that's capable of extracting chemical energy with oxygen is using this kind of protein. Structurally, it has the shape of a lollipop, like an upside down lollipop, if you will. The stick of the lollipop is what's embedded in the inner mitochondrial membrane. This is going to be the proton channel. And the head, or the sugary part of the lollipop, is where the ATP is made. That's here. So the stick of the lollipop is the proton channel. That's what's embedded in the inner mitochondrial membrane. And as the protons flow through that channel is going to physically spin. The potential energy of the proton gradient will become the kinetic energy of this channel spinning, and it works as a turbine. As that channel spins, it's going to drive the production of ATP. And this is a diagram from your textbook that shows that. As the protons flow from high concentration to low concentration, they're going through this channel, this rotor. The rotor spins like a turbine, and that spinning action produces ATP. Now, your textbook has all the right images here and says all the right things, but in my opinion, it just irresponsibly doesn't go the one step further of actually telling you the story. I don't know if this is because they didn't want to step on the toes of the biochemistry components of a curriculum. I don't know why they didn't go this far, but I've made the decision to go just one level deeper into the detail because I think it's very unsatisfying if we don't. Like, still it's magic, still it's a black box how ATP is actually made. So I'm going to go into a little bit more depth here than the textbook does, but I feel I have to. Otherwise, you won't leave this material understanding how ADP is phosphorylated with this kinetic energy. So the stalk portion of ATP synthase, that's the portion that connects the rotor, the proton channel, to the head domain. It has this protrusion. It's asymmetrical. And so the textbook has depicted this correctly. This protrusion here exists. And so as the proton channel spins, and the stalk spins with it because the stalk is connected to the channel, this protrusion is going to spin. And it's going to cause a wave of disruption spinning through the head domain. And this wave of disruption, as this protrusion kind of spins in the head, is going to drive different conformational changes in the subunits of the head domain. And it's those conformational changes that are going to drive the production of ATP. The head domain is actually made up of 3 identical subunits. And each of those 3 subunits can cycle through 3 different conformations. And what's driving the cycling is the spinning of this protrusion. And the name of those conformations are loose, tight, and open. So what happens in the loose conformation? Well, the loose conformation is loose enough that ADP and a phosphate can bind. And then, we're going to squeeze that ADP and phosphate together. That's what tight is for. Squeeze ADP together, squeeze that third phosphate on until we can make that covalent bond. So the tight conformation makes the ATP. That's where the phosphorylation occurs. And then open, you can probably surmise. Open opens the grip and releases that newly made ATP out back into the mitochondria. And then we repeat. Back to loose, bind a new ADP and phosphate, squeeze them together tight to make ATP, and then open to release that ATP, and then back to loose and do it again and do it again. So that means 3 molecules of ATP are made per single revolution of the stalk, and this allows ATP synthase to make hundreds of ATPs per second. So let's dive in just that little bit deeper here so you see what's actually happening. This is a very typical diagram of a biochemical textbook. But even this I'm not completely satisfied with, so I've kind of co-opted this a little bit. So here is the loose configuration up here. Right? This light blue is loose. We're in tight over here, and this is open for now. But we've paused this so that loose is straight up. And in the loose configuration, we're binding ADP and phosphate. But why are we in this loose configuration? Because that's where the protrusion has jostled this subunit. And I'm representing the protrusion of the stalk here by this arrow. So the protrusion is pointing here. We've jostled this subunit. We've forced it into the loose conformation, and it's bound to ADP and phosphate. But protons flow through the channel. As the channel spins, so does the stalk. And as the stalk spins, so does the protrusion. Now remember, wherever the protrusion is in the loose configuration. So this domain here, this region, is going to go from open to loose. But here we've gone from loose to tight, and here we've gone from tight to open. So tight, what happens in tight? Squeeze that ADP and that phosphate together to make that third covalent bond. We've made ATP. But then protons flow, the stalk spins, the protrusion spins, and the conformation switch again. So the protrusion is now going to be pointing in this direction, which means this is going to be loose. If this is loose, then tight's going to flip over here. If tight flips over here, open's going to flip to here. Open is the release of that ATP. And so again, if we just back up, we're in loose because that's where the protrusion is. Protons flow, so loose shifts counterclockwise by 1, but so does tight, squeezing this ADP and this phosphate together into ATP. Protons are still flowing, the protrusion still spins. We still spin 1 unit counterclockwise, and now we've brought open up and we release that ATP. But like I said, we're making 3 ATPs per revolution of the stalk because each one of these 3 domains is functioning independently. I want you to look at this animated GIF in 2 ways. I want you to look at it in any moment of time and kind of look at it in almost your periphery. And you'll notice that each of the 3 subunits of the head are in each of the 3 different conformations. At any one time, one of these subunits is in loose, one of these subunits is in tight, and one of these subunits is in open. But as the stalk spins and the protrusion spins with it, now I want you to pick any one of those thirds. I don't care which one, the one on the top, the lower right, or the lower left, but pick one of them and just stare at it and watch it cycle from loose to tight to open. I'm looking at the top one, so hopefully the narration will sync well enough with the video when Zoom converts it that it'll remain synced. But I'm going to start, and it's on loose, tight, open. I'll do it one more time for the one at the top. Loose, tight, open. And let's look at what happens in loose. In loose, ADP and the phosphate bind. At tight, they're squeezed together to make ATP. And in open, they're released. So the top one is loose, tight, open. The one on the lower right is tight, open, loose. And the one on the lower left is open, loose, tight. That's kind of, they're almost singing it around. And so ATP is constantly being made. And with each full revolution of the stalk, 3 ATPs were made out of sync with one another, but 3 ATPs were made in this way. And just to backtrack, the energy for adding that third phosphate is coming from the tight conformation. Well, what's allowing the tight conformation to form? The spinning of the stalk. What's making the stalk spin? The spinning of the proton channel. What's making the proton channel spin? The protons flowing through it. Why are the protons flowing? Because they were in this huge electrochemical gradient of 1. Why were the protons pumped there in the first place? Because of the energy released by the ETC. What gave the energy to the ETC? The electrons that were delivered there. Where did those electrons come from? The citric acid cycle. What was fed into the citric acid cycle? Acetyl-CoA. Where did the acetyl come from? Pyruvate. Where did the pyruvate come from? Glucose. All of this has come from the energy of glucose. And that's really the story, right? Glycolysis breaks 6-carbon glucose into 2 3-carbon pyruvates. That pyruvate is then transported into the mitochondria and converted into acetyl. Acetyl is linked onto CoA. Yeah, we lost a carbon dioxide along the way. Pyruvate's 3 carbons, acetyl is 2. Yeah, we did some reduction of NAD in the citric in glycolysis. We made some ATP, but it was the acetyl we were after. We link that acetyl onto oxaloacetate to make citrate, and then we break bonds and steal electrons. Break bonds and steal electrons. Make a triphosphate, okay, fine, whoopee. Break bonds and steal electrons. Break bonds and steal electrons, and regenerate oxaloacetate. Where do all those electrons go? Well, the vast majority of them are on NADH, and the NADH electrons are delivered to complex 1. Complex 2, here's complex 2. Complex 2 is succinate dehydrogenase. The enzyme that catalyzed this reaction is this enzyme. It never let go of its FADH2. It still has it. The electrons migrate from NADH onto Q, which delivers them to complex 3. The electrons migrate from FADH2 onto Q, which delivers them to complex 3. Complex 3 delivers those electrons to complex 4 using cytochrome C as its shuttle, and complex 4 delivers those electrons onto oxygen, reducing it to water, the metabolic water that we exhale as waste. But most importantly, each of those electron transfers was a redox reaction that had a negative delta G, and that energy that was released was used to pump protons against their concentration gradient. And now those protons are flowing through the proton channel of ATP synthase, making that channel spin, making that protrusion on the stalk spin, and driving these head domains through loose, tight, open, loose, and open, loose, tight, cranking out ATP through oxidative phosphorylation. The phosphorylation of ATP driven by energy of oxidation, the oxidation of electrons from glucose. Isn't it like amazing? Isn't it just incredible? Yeah, it's like science fiction. And as awake as you might be right now, hopefully you're wide awake and as excited as I am, but whatever degree of consciousness you have, you are conscious because of the ATP being made in your cells, and that ATP is being made by your mitochondria. This is happening in you right now, right now. All right, so let's tie up some loose ends. The energy stored in this proton gradient is extremely valuable. Yes, it can be used to make ATP, but it can be used in other ways too. And now I'll bring up the bears. So if you allow those protons to flow through ATP synthase, but you decouple the proton channel from the stalk, so only the bottom of ATP synthase spins. Well, now the potential energy of that proton gradient is being converted into pure kinetic energy, just like the spinning of the proton channel of ATP synthase. And without any chemical capturing of that energy, the only thing that that kinetic energy can become is heat. So in that case, all of the potential energy of the proton gradient becomes heat energy. And this is the heat energy that allows bears and other hibernating animals to survive hibernation and not freeze to death in the winter. Like, again, amazing. Hibernating animals have a way to decouple their stalk from their proton channel, and therefore all of the potential energy of protons flowing through the channel and becoming kinetic energy is released as pure heat, pure enthalpy, and that's the heat that keeps these hibernating animals warm enough to survive the winter. But that same energy can be used for other things too. Some molecules, like phosphate, like pyruvate, right? Pyruvate needs to get into the mitochondria, and even ADP in order to be phosphorylated. Some of these molecules, to keep up with the amazing efficiency of ATP synthase, can't be relied on for passive diffusion to bring them into the mitochondria. They have to be physically pumped, forcibly pumped into the mitochondria against their gradient. ATP synthase is so good at its job that we need to keep a steady and constant supply of raw material for it to make ATP with. So some of the kinetic energy of proton flowing is used to pump these molecules into the mitochondrial matrix, into the mitochondrial space there, so that they can be used that way. This is called coupled transport. And the best analogy I can think of for this is imagine a huge crowd of people going into a hotel through a revolving door, right? Those are the protons going where they want to go, flowing through the proton channel to get there. Now imagine you get caught up in that crowd. You don't want to go into that hotel, but you got caught up in the crowd. Well, I don't know what to tell you because it sounds to me like you're going through that revolving door whether you want to or not. You've gotten caught up in the flow of a lot of energy of want, and you got brought into the same hotel whether you wanted to or not. Phosphates and ADP and pyruvate get carried into the mitochondrial matrix by riding the wave of protons flowing into that matrix themselves. This keeps a constant supply of the raw materials needed for ATP production flowing to where they're needed. This energy is also used to import mitochondrial proteins that are made in the cytoplasm and need to find their own way into the mitochondrial matrix. They also hitch a ride on this proton wave. So cellular respiration is amazingly efficient. Glycolysis, converting pyruvate into acetyl-CoA, acetyl-CoA into the citric acid cycle, and then what we've talked about here, the ETC and proton pumping, all the way to get to ATP synthase. All of that is very complex and very discrete. Lots and lots of steps, lots and lots of reactions. Why? Well, part of the reason what we've already said. It's to release the energy in that glucose in small enough packets that it can be captured without cooking the cell, without frying the cell for too much energy release. And also with each one of these reactions, we can bribe the universe with a little bit off the top, give the universe a little bit of that negative delta G, and allow these reactions going forward. But despite all that, this is the most efficient way to extract energy from glucose. It's the most efficient way to capture as much energy as possible from the covalent bonds of glucose that we've broken, and stealing those bonds high energy electrons, and then passing those electrons on to NADH. Once this gets to the ETC, it's pure energy. It's electron energy that's being used to pump those protons, and then of course spin ATP synthase. In fact, the amount of energy is quite substantial. Each reduced NADH carrying the 2 electrons that it robbed from glucose, the 2 electrons from either glycolysis or the citric acid cycle, provides enough energy to make 2.5 molecules of ATP. So essentially, each electron robbed has a little bit more than 1 ATP's worth of energy in it. This table, table 14.2 from your textbook, shows the breakdown of cellular respiration through the context of ATP production. So from glycolysis, we make 2 ATP profit from glycolysis directly, substrate level phosphorylation, and then we made 2 NADHs, which allowed us to make 3 electrons. We don't make the full 5 electrons there because we actually lose some of that electron energy because of having to drive some unfavorable reactions of glycolysis forward. Got to bribe the universe with a little bit more of a bribe there. Then the 2 NADs that we get from the conversion of pyruvate into acetyl-CoA, we can make our full 5 ATPs from them. 6 NADHs from the citric acid cycle, 15 molecules of ATP from them. The electrons from FAD, they're a little bit less high power. We can only make 1.5 ATPs from FAD electrons. So 2 electrons on FADH2 from the citric acid cycle, 3 ATPs from that. And then we make 2 triphosphates, right? We run the citric acid cycle twice for each molecule of glucose. So we make 2 triphosphates. So you total that up, and that's 30 ATPs from 1 molecule of glucose when we're allowed to do this entire process, glycolysis to the citric acid cycle, to the ETC to OxPhos. And again, I just mentioned that succinate dehydrogenase keeps its electrons on FADH2. Succinate dehydrogenase is complex 2 of the ETC. It's going to give those electrons to CoQ and shuttle them to complex 3. So they fast track directly into the ETC, bypassing complex 1 completely. And because of that, they're lower energy electrons. And like I said, we can only make 1.5 ATPs per FAD carried electron. Approximately 50% of the potential energy that glucose has to offer is captured as ATP, as the chemical energy of these triphosphates by the end of aerobic respiration. And as much as that sounds wasteful, it's only 50%, you might be thinking. That is by far the most efficient energy conversion that I think we're aware of, period, as a species. Car engines typically have 10% efficiency. So when a car engine burns gasoline, 90% of that reaction is given off as pure heat. Only 10% of that reaction is used as work to drive the car forward. The most efficient car engines we can make, electric or gasoline, are 20% efficient, meaning 80% of the energy in the fuel is lost as heat and 20% is captured for work. And here we're saying that 50% of the energy is captured. So more than doubling even the most efficient car engines that we can make, and 50% is released. Where does the 50% of energy go that we don't capture as ATP? Well, it's sincerely my hope that by now in this course you should know the answer to that. The other 50% of energy from glucose that we were unable to capture and use to make ATP, well, that's why we're running at 98 degrees Fahrenheit. That's why we're running at 37 degrees centigrade. That's why we are so hot as a species. And all of that energy is being released and dumped out from us constantly to bribe the universe, make the molecules around us move more chaotically, increase the entropy in our surrounding environment to allow us the privilege of being so non-entropic and so ordered internally. What a story. What a story, right? So, to summarize what we've talked about here, cellular respiration relies on OxPhos, oxidative phosphorylation occurring at the ETC and ATP synthase to maximize ATP production from glucose. The mitochondria are the powerhouses of the cell because they are the site of aerobic respiration. The most ATP possible is being generated in the mitochondria because of the presence of the intermitochondrial membrane and the aerobic respiration that occurs there. Electrons from NADH are delivered to complex 1. The electrons on FADH2 are part of complex 2, and they're delivered to and transferred through the electron transport chain along the intermitochondrial membrane. The electron transport chain releases energy through subsequent redox reactions. Energy is being released a little bit at a time, and some of that energy is being used to pump protons against their concentration gradient. That active transport, the rest of the energy is used to bribe the universe. Before I go into the proton gradient, just a quick review as well. The electrons being delivered by NADH are being given to complex 1. CoQ brings them from complex 1 to complex 3. Cytochrome C brings them from complex 3 to complex 4, and complex 4 delivers them onto oxygen so they can be released as wastewater. FADH2 electrons are part of succinate dehydrogenase. Succinate dehydrogenase is complex 2. It passes its electrons to CoQ, which brings them to complex 3. From complex 3 onto the mobile carrier cytochrome C to complex 4 onto oxygen. ATP synthase is this amazing lollipop-shaped protein. It's a molecular turbine. Protons flow through the turbine of ATP synthase. Those protons flow back into the matrix and convert ATP into convert ADP diphosphate into ATP. Chemoosmotic coupling is the way that this works. This is what links the energy of proton movement to ATP synthase. Loose, tight, open is the manifestation of chemoosmotic coupling. The intermitochondrial membrane folds to increase its surface area, creating these cistrae or these creases. And this enhances the amount of ATP production because we have that many more electron transport chains and that much more ATP synthase in that larger membrane. And oxygen is an empty trash can. Its sole job is to serve as the final electron acceptor of the electron transport chain. When those electrons has given up all the energy they possibly can, they then give that energy off. Once the oxygen had, oh my, once the electrons have given up all the energy they possibly can, they are now low energy electrons. They are energy depleted, and then complex 4 puts those electrons onto oxygen, and reduced oxygen is water. That water is exhaled as waste. Each reduced NADH provides enough energy to make about 2.5 ATPs. Each reduced FADH2 makes about 1.5 ATPs each, and that makes cellular respiration by far, hands down, unparalleled, the most highly efficient energy capturing mechanism we have out there. 50% of glucose's potential energy is converted directly into ATP, and the remainder is released as heat, why we run so hot, heat that bribes the universe by increasing the entropy in the world around us so that we can remain ordered within it. That's it for this video. Again, I'm recording this on my laptop today, so I'm not quite sure how long this one was, but it kept my interest, that's for sure. What a story. When we go into the next video, again, the next video will be much shorter. We're going to go into some of the intricacies of the ETC. We're going to talk about why these ETC redox reactions are favorable, why they're spontaneous, and get a little bit more comfortable with what's actually happening to the electrons as they move through the ETC, because it's fair to say we focused on the protons today. But until then, and as always, thanks so much for watching, and please let me know what is confusing and what you would like to discuss in class with the dialog box that's going to pop up in just one moment.